One. Hello!
Hi there.
Welcome to YouTube Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to enabling the public with the tools of biotech.
My name is Fazal Lam, and I did my PhD at Imperial College London, where I made flesh-eating bacteria glow in the dark. But since then, I've also worked in research integrity, and currently I'm an editor at a scientific journal.
Every week, we meet to talk about microbiology, and today it's our deep dive week, where we look at each figure in a paper that we chose last week to learn and criticize the scientific findings. This is a journal club, and we encourage our audience to leave us questions and comments.
And next week will be our news week. So if you like what we do this week, next week we're going to be going through a number of different news stories, paper by paper, and then we'll choose what we'll be focusing on, like we do today, in much more detail.
Yep, you can follow along with the papers that we discuss on either week in our shared Zotero library, linked in the doobly-doo below.
Yeah, and if you want to, we want to hear from you. So please use the comments to comment at us or tweet us using the hashtag #MicroTWJC hashtag.
So this week, we chose directed evolution of potent neutralizing nanobodies against SARS-CoV-2 using CDR swapping mutagenesis.
Yeah, so this is interesting because nanobodies are a big thing, but well, they're not a big thing. That's the opposite. They're a smaller thing.
They're a
like,
small thing.
they're antibodies, but they've got they can go places where maybe another antibody can't go. They're small, so they're easier to make. So there's been some interesting developments with them for like fighting SARS-CoV-2. And this paper is about how to build a better nanobody.
Yeah, I think I remember in one of the previous news episodes, we covered something where it was like nanobodies used for intracellular pathogens. I feel like I said that last time when we in the news where we chose this paper as well. And I
Yeah.
mean, it's just interesting. Like they are much smaller molecules. They can potentially thus go places that like other molecules couldn't. But then they're also easier to make because there's less material, less biological material to have to output.
Yeah, and also like I remember like reading a while ago there was like someone trying to create an air spray, kind of an inhaler where you can inhale these nanobodies and use that to fight SARS-CoV-2.
Fascinating. Yeah, I guess like
Yeah,
smaller also means easier to make small particles of. There's all these different things that come.
yeah,
So this image that we have pulled up for you guys,
yep.
this is a picture from the PDB. So that's the protein
Database.
data bank. They have structures of all sorts of proteins, and they have a structure here of an antibody versus a nanobody. So everyone knows those antibodies are a Y shape. They're actually made of uh, they're a dimer of two different two things that are made of two chains. So there are four components in an antibody. There's the heavy chain, which is the longer chain that starts from the tip and goes down to the other tip, I guess, the
Yep,
stalk.
those are in blue.
Yep. And then there's the light chain, which is in green, and those are just at the tips, the two different tips. And it's those Y tips of the antibody that do the recognition that bind to different things. They actually have sort of flexible regions at the tips of those stalks that interact with different molecules.
Yeah, and actually fairly recently discovered that animals like camels, alpacas, they have and something in addition. They have these nanobodies they produce in addition
Yeah.
to this, which are much smaller. They
Yeah.
only
So
have
they don't have the light chain, and so they only have the chunk of the their recognition zones are a little bit smaller, but they still work. They still recognize things.
Yeah, and like there's been quite a lot of interest in them because also they're less immunogenic so that you can use them.
Mm.
So and also like there's been interesting because like to run these essentially to create one of these nanobodies you'd have to inject it into a big alpaca and then wait for it to produce the antibodies. Um,
Yeah. Oh, I was gonna say that. Yeah, that's an interesting point. If you're saying that they're less immunogenic, I didn't know that, or I hadn't heard that at least.
I mean I've heard
Um,
that, but I mean again I didn't
yeah,
look
we
into the
don't.
papers to delve into so
Sure,
that's
no, that's
yeah
very true. But a good point. But that's a good point to bring up that when and this is a point people may not know about biologics, right? Like that's a class of drugs that's very popular these days. They're biological molecules, right? Antibodies and nanobodies, they're biological molecules. And so when you inject that into a person, your body may be like, oh, a foreign protein. Like we need to recognize this and make antibodies towards it. So you'll get antibodies recognizing the antibodies that are recognizing the thing that you want to deal with in your body. And that can reduce the effectiveness of the drug. A really common story that's related to that is hemophiliacs, right? There's a lot of biologics for hemophiliacs. They have to take those antibodies a lot, and those antibodies are specifically like tamping down a specific like blood reaction. And so, or sorry, they're giving them exogenous molecules, right, of this clotting factor. But their bodies aren't adapted to knowing that is supposed to be around. And so they get a certain percentage of people will get antibodies against those clotting factors.
Right.
So this is, your body is naturally attuned to making sure that like the balance of what it knows is what stays. And so that would be a very good thing if we could confirm, I guess, that nanobodies were less immunogenic, because this is something that all biologics face in therapeutics.
Yeah, I mean, on the other hand, it's easy to say something isn't immunogenic. We haven't tested it for a very long time, so it could be that something comes out. Because I remember like hearing this a lot about like certain gene therapies early on, that using a virus that isn't immunogenic until we test it in enough people, and they go, well, it's actually slightly immunogenic. So it's one of those.
Yeah, that's true.
So
yeah, because now we've had like the biggest test, right? Like if people said that AAV wasn't immunogenic, but now AstraZeneca, right? Like now we see a vaccine and we're like, actually, yeah, there's some immunogenicity.
Yeah. So I mean, I guess I'm gonna torpedo my own statement that I just said, saying that, oh, this isn't immunogenic. And I thought about it for a second. Then actually, like, can I really say that? Doubt. We're all about doubt in this show.
Totally. Yeah. No, it's good. It's good to show like that's. When we did our PhD trainings, right? Like you always want to say the limitations of your techniques, but also your knowledge, just so you're getting that clear communication.
It's much easier when you point out your own weaknesses before your supervisor does. So it makes the lab meeting so much easier when you go, well, I'm sorry I made all these mistakes. And I'm like, why did you make... Well, now you know you made these mistakes, so don't do them next time. I'm like, okay.
But we're humans. Mistakes happen.
Yeah.
All right, let's see what we got next in our intro.
So yeah, so talking about like how the actual antibodies are created normally, it's usually through B cells create antibodies, and they select for binding affinity towards an antigen. And in this process, the genes that make the antibody go through this thing called somatic hypermutation in this germinal center, where they organize and pick out the B cells that produce the right antibodies based on the binding quality of them.
Right. Yeah, we've talked about this process, I think, twice now in deep dives. Once we talked about it in the context of germinal centers, that's the place where antibodies mature and how that gets disrupted by SARS-CoV-2. And then we've also talked about it in the context of vaccination and how B cells, like, I mean, we want to tickle them in the right way so that they make those antibodies that we need. And so, yeah, I just want to remind everybody that that's how we get our antibodies, right? There's this huge diversity of B cells in our bodies, and we have to find in that diversity the right B cell that's going to bind to that antigen. And then our bodies use evolution, or something called somatic hypermutation, to make those antibodies even better binders.
Yeah, and the interesting thing about this is like how it's done naturally. But in this paper, what we'll be seeing is how we try to replicate this process in a petri dish without involving an animal where we actually do the direct mutation in the lab.
Absolutely. Yeah, which is like it's just crazy, right? Like how far science has come, where it's like we used to make antibodies by injecting, we still make antibodies that way, by injecting animals and having the animal make antibodies. I don't know, you probably work with a lot of rabbit reagents, right, in your labs. Yeah.
But I mean, another thing that is also like kind of a blocker, say if you got an antibody, you want to create an antibody for something like actin, or something that's very common in all animals. Most animals won't create antibodies against them because they're trained not to recognize self. So there is a limit to the kinds of antibodies you can create using this process, simply because the animal doesn't want to create autoimmune antibodies. So if you want to like have a marker that'll like kind of attached to the heart or anything like that, it's going to be very, very difficult to create that in an animal.
Yeah, good point. Oh yeah, let's go to the next slide then.
Yeah.
Um, so yeah, we have this process of directed evolution, which is essentially the same thing. You have to have a really diverse set of different targets, and you have to have a way to pick out from that group the ones that are the best at doing the phenotype, the thing that you're interested in. And then if you repeat that process, oh yeah, this is great.
Yeah, so I'm just going to focus on like the different ways you can generate that kind of. So firstly, you've got the generating the genetic diversity. So you've got strains that have mutators that can cause mutations to happen. You've got error-prone polymerases. So polymerases' job is to take DNA and replicate it. But sometimes if you get polymerases who are a bit like bad at their job, they'll introduce mutations. They're not, yeah, they're not good at copying stuff. They're trying to copy stuff down, but the handwriting's a bit too squirrely, so they end up like transcribing a completely new gene accidentally, or just adding in a little mutation. I mean, chemical mutagenesis is similar to that, where you've got like things that look like bases that aren't, and then that confuses the polymerases, so they end up putting like kind of you've got like essentially a gap in the genetic code where you can put anything in there, and that's one way to introduce a single mutation. And also DNA shuffling, where you take DNA strands and just use PCR to bind them to different things. So often you'd have like say ends that are complementary, like your polymerase will be linking them, and they'd end up like seeing two ends that are the same, and they think, oh, this is part of the same gene, but they're not, and it'll just get confused and just transcribe the whole thing, and you'd end up fusing different genes.
Absolutely. That's like combinatorial almost, and we'll see that actually come back up in this particular paper.
Yeah, in this particular paper, I think things we need to look out for: error-prone polymerases and DNA shuffling. And
so that's the way that we make the diversity, or like get all those variants. And then you have to move to the next phase where you're selecting for a phenotype. And so that can be done in many different ways. Here they don't really tell us too many details, but in the body, right, the way that that's done is just through binding, right?
Yeah.
If the thing that makes, you have your gene and it's making a particular molecule, then you see, does that molecule bind, an antibody in this case, does it bind to the thing that I'm curious about? And then you wash away everything that doesn't bind, or in the body's case, you don't stimulate the things that don't bind. And that's a way of reinforcing this is what we want. We want this particular thing from that huge diverse set.
Yeah, I mean, here, this doesn't assume that it's towards binding. This could be towards any mutation. So they look at various screens to pick out mutants. So usually you'd have a reporter or something like that. And then from there, they'd pick out like whichever kind of mutants that isolate each mutant. And they're trying to figure out what's in there. And then you can put it through another round of diversification.
Yeah, you have to then remove the genes from that particular scenario again, and now the pool of genes is enriched for things that do the thing that you're curious about, and so then you mutate them again, and you pass them through the cycle over and over again. And the idea is that because you're selecting, because you're the natural, well, it's not even natural selection, you're the artificial selection, right? You're the environment that's selecting this gene pool, you're getting that quality that you want. But it's not super straightforward, because like the design of that selection process is, there's oh gosh, I hate that word. There's more than one way to skin a cat, right? There's more than one way to sometimes achieve that bar that you're setting of selection, right? Like for example, if we're looking at binding, doesn't mean it has to bind specifically, right? So you have to really be careful about the way that you design your selection test. Otherwise, you might end up selecting for something you didn't mean to.
Yeah. So yeah, the actual the way you select is so important, because you could end up, so if you're selecting to bind something, you could end up selecting something that binds to everything, and you end up creating the universal glue, which isn't exactly what you necessarily want.
Yeah. But it's a fascinating technique. I mean, this is, I think, 2018's Nobel Prize went to 3 people for directed evolution. So like this concept is a very powerful technique, and we've also seen it. My goodness, we've covered a lot of papers now. We've also seen it in a previous episode where we talked about ACE2 decoys.
Yes.
And so that was like they were making these molecules that were decoy ACE2 to suck up all the spike protein and confuse. Oh, actually, yeah, I picked out a thing. They designed these different scaffolds to bind places on the SARS-CoV-2 receptor. So they're like ACE2, but they aren't ACE2, right? And they were soluble. And they actually ended up using directed evolution to find things. They had all these candidates, and then they evolved them to find the point mutations that would make them more stable and better binders.
Yeah, so you can see they've done some DNA some shuffling of different genes. So here they've got like various combinations, and this is a lot to take in with the eyes. And but
Yeah, this is so. I mean, this was a weird one because they actually used their directed evolution after they did their like binding. They found the binders through computational methods
right.
first, and then they used directed evolution afterwards. So it's almost like there's different like computers can predict what binds, but when you really do that directed evolution and you see like what's actually happening, you get this. There's more of a correlation, I guess, to reality in those types of experiments. And they ended up taking those mutations and re-engineering them into their designs. So they didn't use their evolved versions per se. What they did was they studied what they evolved, and they were like, okay, we see these patterns coming out. Let's take those patterns and put them into what we were designing.
Yeah, so you're still using evolution, but in this case, it was using the information it produces. So you see how like
Yes.
a shark and a dolphin are very similar in body plan. And you go, do you know what? I can build a better version, but I can take out what's similar between them in order to create my perfect submarine.
Perfect. Yeah, no, that's a really good analogy to how it was used in this context. So I think that should highlight the importance of directed evolution. Like it's not just that you have to make the thing that you want, is that we're also querying biology to say, I don't want to have to think about how it's done. Just like set up the conditions and show me nature, right? Like you find a way, find a way
Yeah,
for me.
let nature compute it for you if you can.
Yeah, yeah.
And that is essentially what directed evolution is doing in these cases.
Hmm.
We're running this through an algorithm, but the algorithm is using like kind of biological bits to do everything.
Yes,
And
yes.
it's a good simulation because you're using the actual components that you'd be
Hmm.
so in this case they'll be using like the actual SARS like receptor binding domain and actual antibodies and then iterating them. Uh,
Right, right.
yeah. So yeah, so you talk about
So
sorry.
oh, this is just like a previous work has been done in this space. So, SARS-CoV-2, popular subject.
Yep.
Many papers have come out on it. In 2020, a group published that if you immunize alpacas to get those nanobodies, and then you put them into a directed evolution experiment. In this case, they use something called phage display, which maybe we'll talk about another time. But they use directed evolution to find better binders from the pool of antibodies that came out from an alpaca. And what they found was, oh wow, you could focus the antibodies that came out from an immunization to stronger binders. And they found a candidate TY1, which you'll see is a sort of a positive control that they'll use in the paper that we're looking at today.
Yeah, and I think another thing that we should probably highlight is like the actual like way that the nanobodies bind is using this complementarity dependent region, which is essentially the region that actually does
Yes.
the binding to it, and that has like three
Hmm.
main components, which are called CDR1, CDR2, and CDR3, which is pretty easy to remember, right? Uh,
Yes.
yeah, and these are the actual parts of the kind of like if we're going back to the lock and key thing that everyone goes through for enzymes, like this is
Hmm.
the actual kind of bit of the lock that'll be gripping onto the key. Uh,
Yeah. Yeah, they're generally, I like to think of them as like they're kind of mushy, right? Like they're
yeah,
usually flexible loop bits that stick out from the tips of these proteins. And then yeah, there's so many different variations that you're sort of bound to find one that just mushes into the the thing that you want to bind to.
yeah. And this was like based off alpaca, but I think in this one they're actually looking for a faster way to do it and doing it entirely
Yes.
in vitro.
That's right. And so, yeah, they don't want to have to take the pool of things that come out from an animal because then you have to get the animal, you have to deal with animal stuff, right? Like that's more difficult.
Mm.
They think it might be faster to just like interrogate something else and they'll use a library. So their diversity has been prepared already for them in this 2018 paper,
Yeah.
where they took a common framework and then they varied those CDR regions, just using, I think, error-prone polymerase was the way they did it, and then they created this huge library of all these variants. So in A, you can see like the different variation constraints that went into the different regions, and then right, that star shows you that that star region could be all these different components, right, that they put in. And then B is like, that's a nice visual to show you how those CDR regions are sticking out from the top, right? So those, when they get varied, it means that that surface is getting varied. And then you can see in C, they actually looked at like whether or not they got the diversity that they wanted from their variants, and they were able to.
Yeah, so this is this is great because also like takes back control of the process from the alpaca. So we're
Yeah.
because because when you're using an animal to do this sort of work, you are reliant on the animal's immune system doing everything you expect of it, and that's why kind of when they did the kind of directed evolution after they ran it through the alpaca just so that they could get it better. But now doing
Yeah.
entirely synthetic, I mean, also like from an animal welfare perspective, the less you have to use animals, the better. So,
Well, Foz, actually, a point that you brought up earlier is another reason to move out. When you were saying that some animals won't make an antibody that's directed to an antigen that's too similar to theirs.
yeah,
Right. And we don't really know, like, what, if we're just trying to make different antibodies out there, we don't know, like, what incompatibilities exist, right, between that antigen and an animal. Or rather, it's very difficult to figure that out. And so this sort of sidesteps that problem by saying, we're not going to worry about the diversity inside of an animal that we're selecting from. We're just going to deal with an unbiased pool. And they call this a non-immune library. That's the term that comes with this.
and so they to begin their directed evolution experiment, so they followed this method where they'd isolate their candidate and then separate it using magnetic activated cell sorting. So with that, they'd have these beads that are like these magnetic beads are covered in something that would bind the antibody. And then you have this, well, brilliant, because I use an equipment like this where you'd basically put a magnet on it, and all the beads would just stick to the magnet, and you could wash off everything else. So you capture quite a lot of binding, and you can capture, like, say, if you got yeast that's expressing this antigen on its surface, as shown in the left-hand side of this, then that would then all the yeasts that are expressing it would be saved, and then you've got all those genes that produce it, and then the next step would be to
in here, the VL and the VH, that's the light and the heavy chain.
Right.
So this was not a nanobody system they were using, right? This was an antibody fab system from 2006, Chow et al. And the idea is that, it's expressed on the surface of a yeast, anchored by this protein, AGA2P. And so every yeast has a different fab that's anchored to its surface. And then, as you say, you're using these magnetic beads covered in antigens to pull out, right, from that yeast population, the yeast that contains the fabs, or in our case, the nanobodies that bind to the antigen of choice. And that's the phenotype selection, right, step in this experimental evolution experiment.
And I guess we should talk about like why we're using yeast. No, went up ahead a little bit. But yeast, they're so usually we've talked about like using E. coli, and I think we've done some phage display. But the thing about yeast is they're eukaryotic cells, so they're much more similar to human cells. So if you're creating something like an antibody, a yeast is going to be a lot closer to the structure you'd find in a real human, rather compared to, say, if you grew up in a bacteria. So it does a lot of very complicated things. I think there's a lot of stuff with disulfide bridges that yeast can do that aren't necessarily easy for bacteria to do. So
right. And I mean, in this case, we're not actually emulating a human protein. It's like yeast are also more similar to camels than they are to bacteria.
I mean, bacteria. I mean, I don't know your source for that statement, but I'm going to be looking at camels very differently now. But I mean, yeah.
Well, no, I
So
mean it's just the eukaryotic thing, right? Like
yeah,
humans and camels.
humans and camels. So they're equally related to yeast.
Yes.
But yeah, yeast are a lot easier to culture compared to human cells, because human cells are
Mhm.
finicky and they're very fussy, whereas yeast cells are a lot more easy to deal with.
Yes. Yeah, and there's so many tools, right? And this
Mm.
is just an example of it, right? Like in 2006, this tool existed for screening fab fragments on yeast. So like, let's use that to screen through these nanobodies.
Yeah, and they take these nanobodies and they anchor them to surface through this yeast surface protein called but other surface proteins have been used.
Hmm.
So that brings us to the first figure S1, where in the previous study they used a GPI anchor,
Yep.
which I guess was good for that study, but the AGA2 is what is usually used. I think that's what the impression I got.
Yes,
Yeah.
maybe. I mean, the study was, yeah, I don't know why, because that's 2018, right? Maybe they just didn't have.
Mm.
It's all just about selection, right, of the system. They
Yeah.
probably just had the platform in GPI 1. But yeah, so what you're seeing here is flow charts,
Mm.
which we've seen before, right? That means all the cells are being passed through like a tiny stream and like interrogated with a laser, right? That laser is saying, oh yeah, it has this thing on it or it doesn't have this thing. And so in the control, meaning like there's no staining of that particular thing, you get what the cells look like without any staining, right? And so that's a single peak. That's good. That's cells. But when you stain them using an antibody, stain I think they use MYC tag or something like that,
Mm.
you can see a second peak forms. And in the AGA2 row, that second peak is much more well defined.
Yeah.
And so that means that there's a lot more cells there. They have just more of the protein that you want on the surface. And you compare that to GP1. Okay, there's still a peak there, but it's a little less defined. It's a little lower. There's just fewer cells and they express less.
Yeah, so if you want to, like, say, scale this up, it's a lot easier to take that big second peak and say, okay, these cells definitely express it. Whereas in the first one, it seems to be a bit messy, a bit more variable, and so if you're looking at it from a long-term perspective of like, I want to create lots of nanobodies, you really want a big second peak that has a defined population of
Yeah.
yeast that produces the protein you want.
Mm-hmm.
So
Totally.
much easier to filter out. Um, which brings us to figure one, where they express an existing that existing library that we talked about earlier. So
Mhm.
again, we've got this talks about the first step, which is magnetic activated cell sorting. Then you get your first FACS sort, and you've got your expression of nanobodies. So that again, you see those two peaks, but now it's almost like in 3 dimensions, where you've got your
Yeah.
antigen body binding and the nanobody expression. Because the
Mm-hmm.
S1 showed us the ability for the cells to show nanobodies, but this one actually goes, okay, well, can these actually bind the RBD? So,
Yes, yeah. So in this fact sort, they're also staining essentially, yeah, with that's attached to the FC receptor. So like they have now, so you'll get both, right? They use an antibody to stain to see the surface expression that gives them nanobody expression, and then they use the soluble and tagged receptor binding domain to give you the y-axis of the antigen binding amount.
yeah, so when you're reading these graphs, you have to look at those and you think the sweet spot is going to be in the top right corner. That's where
Hmm.
we want to see lots of nanobodies in if we've got good antigen binding. So they
Yeah.
do like subsequent
So there's a lot
FACS
that doesn't
sorts.
work, right? Like when you're
Yeah,
looking at that graph, like there's a lot of stuff that is just junk, but like there
yeah.
are things in the top right or yeah, top right quadrant. And so those are the ones that they take and then they put it through the sorter again to keep purifying yeah.
Yeah, and they eventually found a set of antibodies that were quite good at binding, and they generated them. So there's Ka1 and Kc3. Um,
Hmm. Yeah, so I just want to take a pause there. So the cells that, right, they're sorting this huge population of cells and they're
mm,
continuously choosing from the quadrant that have the most ability. And then from that, they take they blend it up and they find, right, or they actually, I think they probably sequence a single one, right? They grow it and then they find the sequence,
mm,
right, of the best thing in that population.
right. Yeah, that's right. And this is the result. So Ka1 and Kc3, those are the two that came out of this. So these are essentially from that library that we talked about earlier. And next step is to use error-prone PCR to diversify them further.
Yeah. So they're basically so they started with a library that was made to be unbiased, potentially over like an entire space, and now they've narrowed it down to find, oh, these are the ones that are pretty good. So they're going to put it back into diversification to give us two more libraries, each derived from the two best hits, and put it through FACS again.
Yeah, and based on the way that it's structured, they're not just generating like kind of individual errors. I think they're also swapping as well in this at this stage.
It could be. I mean, you could do all sorts of things, right? Like this error-prone PCR, it's going to make changes all over the place.
Yeah.
Yeah, it's making changes all over the place. There's no explicit swap here, right? Like they're not using a technique that does swapping. Error-prone PCR just drops changes across the entire protein.
Yeah.
Like I think, yeah, yeah.
Yeah, and so they ended up having yeah, they end up getting purifying and finding even more like kind of I'm again, I'm not sure where the swapping happens. I'm not sure they necessarily did it on purpose.
They didn't do the swapping at first.
Right.
Right? It's later in this paper they'll do the swapping. So like here, this is just like this is the directed evolution thing, right? Like this is what you would expect from a standard directed evolution experiment. We'll see at the end of the paper where swapping comes into play. So yeah, so from this you get they get a bunch of candidates, evolved candidates. They're very good at binding. They're better than the TY1, which we mentioned before, which was an evolved candidate, but coming from a pool that started it in the alpaca.
Yeah, I mean, you can almost see that here, where you got the TY1. It's got like kind of this line going up, but it doesn't go up quite as far as the other. So you want so for antigen binding, you want to see it go up as high as possible, and also in cells that have high nanobody expression. So basically, as far as the closer it gets to that top right corner, the better. So it looks like KA1 is doing pretty well, but also KC3 and the KC3 derivatives seem to be doing well as well. So
yeah, absolutely. And so already from this, from just them doing this experiment, we can already see the advantages of using the synthetic over the the actual immune one, right, from an animal. But I guess, I just want to say,
sorry.
That it could also be, though, that the technique of error-prone PCR is better than phage display.
Yeah, that's true.
So, like, I don't know, right? That's a hard comparison to make. But anyways, that's all. Yeah. So they sequence, right? They sequence the things that have come out of their evolved, their evolution experiments, and they find that there's not a lot of variation in CDR1 that occurs, but maybe they get they see a point mutation in some of them, but they do see a bit more variation in CDR2 and CDR3.
Yeah, I mean, you see like CDR3 seems to have like massive differences. Like there's some mutations that show up a lot. So there's that A that turns to an N in all the other mutants. And yeah, I think the thing I like about this is that the FACS kind of technique does replicate almost what's happening in the germinal center, where we are picking things based on how well they're binding, and that entire system is like replicating what's happening there.
Yeah, I mean, like I do like this. It's like it's just a fascinating concept that we're essentially the whole directed evolution thing with antibodies is replicating what the vertebrate immune system does normally. But we have control over it, and also we don't have to worry about things like autoimmune antibodies at this stage because, right, that's not we can work from a pool that doesn't have that constraint.
Right. And now figure says 3 plus 3a. So, this is interesting. So they purify those.
So they want to test. So they've made this on yeast, but now they want to make antibodies they can actually test in like neutralization assays and stuff. So go ahead.
Oh yeah, no. What was I going to say? Yeah. So they found that they were able to isolate them at fairly good purity, but also like at this stage you are seeing like how much better the antibodies they've chosen are doing. So this is essentially like looking at how well they inhibit. So I think this case they're looking at a version of SARS-CoV-2 that's created like not the RBD that's expressed on the lentivirus construct. So it's a pseudovirus.
That's right. Yeah, this is pseudovirus.
Yeah.
Okay. I do want to speak to this. That they made so we said nanobodies, right? That's the library was made in nanobodies. it sits on the yeast, so it's just the one little region. But what they did when they made these is that then they took those two nanobodies and they fused them to FC, and they essentially reconstitute a full antibody, right? That doesn't have the light chains because it's the nanobodies as the stocks, and they make that in hex cells, right? So they've actually switched now to make them in human cells. I guess that's just because cell type is optimized for making antibodies. Yeah, I didn't read into that, but like they've switched systems now. So they're going to talk about a bivalent version of the nanobody, right, and the monovalent version of the nanobody. So it's important, I think, to remember bivalent means they've taken the sequence of the two nanobodies and they've like reconstructed an antibody from it, and that's the bivalent form. And then the monovalent form is sort of the easier way to do it. It's just like what the yeast has on its surface.
Yeah, and we're seeing that here they've got these are the bivalent antibodies, and we're comparing them against, say, the wild type, so we've got an actual antibody which is CB6, and the nanobody that was purified in the previous experiment, and they're kind of at the same level of neutralization, and then these directly evolved nanobodies have got like they're a bit better, so it looks good.
Yeah. I mean, I would say it's like looking at those IC50s, that's an order of magnitude more.
Yeah. Oh, yeah. No, you're absolutely right, because I didn't notice that it's a logarithmic scale. So actually, those are quite big differences we're seeing there.
Yep, absolutely. Yeah. So this is a great technique. So it seems, and that holds even when they use live virus.
Yes. So this is this graph shows live virus, and they take, I think, one of the better candidates, and they compare it, and it does dramatically better than the other thing. So.
Yeah. Well, I think an interesting thing, too, that we're seeing here is that it takes a lot more to neutralize live virus than it does the pseudovirus assays.
Oh, yeah. Yeah, I didn't notice that, but you're absolutely right.
Yeah. Which is kind of wild. I mean, it kind of goes along with the stuff that we've been talking about with live virus, that there are more receptors than just ACE2 at play here. And so selecting this particular thing, just blocking receptor binding domain from binding to something like that, it might not be the only story that we're seeing with infection.
Yeah. So, okay, so now they looked at the evolved monovalent nanobodies and anchoring them to yeast cells to show just like their binding patterns.
Yeah, it's really the same thing. I actually think that the reason why they've done it now with the monovalent is because they want to show that like they can also screen like the relevance of like because you'd assume that the therapeutic that you end up making is probably not the monovalent form, although I guess I don't know all those immunogenicity things. There might be other concerns to make, but this is just to show that you can just use the yeast cells themselves that have like the monovalent, and it still works in these. Or actually, these aren't neutralization anymore. Just like the binding of them, you can measure, and it does really well, and it stacks up super well as well to the TY1. And then here they have a different antibody candidate, VHH72. But again, just another monoclonal that's out there that binds to the spike.
I mean, it's really interesting that the way I'm looking at it is also interesting that the bivalent antibody seems to be at least the same measure. No, they're EC50. Sorry, they're different experiments. So
they're doing it a bit differently. Mm-hmm.
So for a minute there, I was thinking, well, they bind it, but no, one of them is actually looking at the in vitro kind of like, can it protect cells or not? So that look. But again,
but then they do. Yeah, but between A and B. KD and sorry, that's the exact same thing you said. Sorry.
Yeah, no, it fooled me for a second. I was about to say, wow, the bivalent is doing much better than the monovalent, but I'm like, no, they're different tests, so actually that wouldn't say the same thing at all. And we've got figure S4, which shows the different a
control. Or not just a control, because controls are very important. It's a control to make sure that the differences that we're seeing in these different binding capabilities, it's not because some yeast cells have more of it on the surface than others, and they're all approximately the same.
Yeah, no, that's a very important thing to make sure, because if those controls were out of whack, then suddenly we'd be telling a very different story. because
Yeah.
otherwise we're taking the fact that nanobodies are easier to construct than antibodies, and then we'd say, okay, maybe that's the reason why like they're doing better.
Yeah, or just like even just within a yeast strain that harbors the particular nanobody, maybe for some reason it's just better expressed on that yeast, right? Like if you remember from the flow charts, there's a huge variation of what's expressed on the surface. And so it could also be that that's driving this particular effect. And so, yeah, they have it here. But they don't, sorry, I just want to say they don't have to do that control for the bivalent because in the bivalent, they're actually like secreting out that antibody and they're purifying
Yeah.
it. Right here, it's because shorthand version, they're using the yeast cells that have the nanobody on it to do these binding tests.
Right, that brings us to S5, which is let's see what we got here.
So
So
I can go. In
yeah,
this one, it's known previously that some monoclonals that bind to SARS-CoV-2 antigens also bind to SARS-CoV classic SARS antigens. And so they're going to try to figure out, they know now that they have this antibody, it binds well, but they want to figure out like how, like where exactly, they want to get some information about where. And so one of the ways you can do it is to look at, does it bind an antigen or an epitope on classic SARS? And you can see the VHH72, it does bind the classic SARS, right? In the open,
yeah, we
in
can
the
see
red, in the red,
yeah,
not the open, the red circles. But the evolved version, it doesn't bind the SARS-CoV-2. So
yeah,
whatever, or the classic SARS. So there's some epitope that's unique on the SARS-CoV-2 receptor binding domain that our evolved version is going after. And that's not what we see in this monoclonal.
yeah. So that kind of narrows down the structure a lot because now we know that it's going to be, whatever it's binding to, it's definitely not been seen on the original recipe SARS. And I think the next
Yep.
slide going to go a bit more into a bit more detail of trying to figure that out.
Yeah, so they'll do it through competition. They pre-incubate the receptor binding domain with a bunch of different monoclonals, as well as ACE2, right? And those other molecules will bind the receptor binding domain at a specific point. So they have these different classes, I guess, that they're calling these epitopes, class 1 to 4. And then ACE2, of course, that's the actual receptor binding surface. And then they measure, and then they take that mixture and they say, okay, how well does this pre-bound mixture bind our nanobody that we made? So what you're seeing on the graph is how well it's binding the nanobody, and then the lines are indicated by
Yeah.
the thing that they're using to compete.
So, for instance, in blue, you've got like the actual as a control, they've got the KCEP3, which is they're going to be using. So that's like their baseline. So you have to do better than that in order to be considered like a good competitor. So the idea is that if I guess both can bind, if it can like tear them away, if it can bind still, even though these are still bound, there's binding somewhere different than those antibodies. So
That's right. Yeah.
so for instance,
Yeah, if you get signal, it means that it doesn't matter. It's not competing. But if you get a reduction in signal and you're going down, right, then it's in competition.
yeah. So for instance, for the purple, the purple, the orange, well, the various purples and the orange, it seems that they're competing, so they might be hitting the same site because they isn't very much. Whereas like for the VH872, we already know that's hitting a different site, and then we've got these other antibodies that are hitting different sites. So thus we get more of an idea of where it's not hitting based on these competition assays.
Yeah, we have this idea that whatever the epitope is that the evolved nanobody is binding to, it's similar to the one that we got out of the alpaca, right? Another directed evolution experiment. It's similar to class 1 and class 2, whatever those are. I guess we could have picked up, the torso or whatever. It's some region on the surface.
Oh yes, I recall those great pictures. They basically like to classify the spike protein in terms of either a squirrel or a torso shaped, and they indicate. I mean, those are wild papers we looked at a while ago, but yeah,
Just like a very funny analogy to see in an abstract.
yeah, it's interesting to see. I think what's happening with ACE2. I think they suggest that it's binding part of the ACE2 binding, but not all of it, perhaps, and that's why you've got this strange like kind of pattern with the ACE2 receptor
Yeah,
binding.
so it's like only at like high concentrations of ACE2 does it seem to like have a lot of competition. That's what I'm seeing.
Yeah. So yeah,
Okay.
So figure S3. I think this
So
is
I think that's sorry, that's as close as they're gonna get to showing us where it binds.
yeah.
Right? Just this general sense that like, okay, it binds something unique to SARS-CoV-2 in this class 1 or class 2 region. And we could probably dig in the literature to find you a better example of that, but actually that information doesn't mean that much to us. Just to know that they figured out a place where it binds, and if you wanted to know, you could dig into the literature, you could find it too.
Yeah, you can probably do some footprinting based on the antibodies that bind and don't bind to narrow it down. But
Yeah.
we don't really have the time to do that at the moment. So
For you guys, yeah.
yeah.
I mean, it like requires some skill, right? Like to make a figure by yourself, but the data is out there, right? I think we talk about this a lot. Like the fact that the research papers are a representation of data that's being done, right? It's a way of them communicating a story to us. But if the data is out there, then you can also take that data and you can like draw your own conclusions. And we've seen that, like one of our viewers like does little snippets, right, of like people's talks, bringing papers together. And the COVIS, when we looked at that, they have a group called Refigure, where you can go in, you can remix figures together. It's a story from them.
Wow. Yeah. I'm trying to think of the most crazy story I can get away with, and I'll go into there and try and prove aliens exist using them. I don't know, but
But maybe, right? Like the world is big. The world is full of data. Like as scientists, we interpret it to try to like tell us something about it. And really, I mean, it takes a lot of work sometimes because you really have to read the background of all that data that you're using. But if you do know it, then you can do that analysis yourself.
yeah. So we're not assigning you homework, you viewers at home, but just in case you.
If you want homework, just let me know.
If you want homework,
I can assign you a task.
Oh. So yeah, now it comes to the point of actually looking at the drugability of these. I think is what they're trying to look at.
Are they good? Yeah, are they good drugs? Can we make them into drugs? So we saw this already. This is just the SDS phage gel of this bivalent FC fusion, right? A reconstituted nanobody. And the idea is that if there shouldn't be too many other bands
right.
on the thing, and they have it in the heated condition, right, and a reduced condition. So the reason why is because if they don't get a band in the heat condition, it means that the heat globbed up them all together, right?
Yeah.
And it removes the band. So it's nice that there's a clear band and it's not like at some ridiculously high kilodalton level from the heat, right? It's stable, heat stable.
Yeah, I think that's important because again, if it starts clumping up, then that might be a bad thing for storage or for other sorts of things. So having some kind of stability is quite good. And
then in the reduced version, they see 2 bands. I think that's because it breaks disulfide bridges. Reduced. I'm not sure why there's 2 bands, but like again, it's not a big clump and it's small and it's bigger. Maybe that's just like a regular version.
It could be the
that's
linear.
It's heat and reduced is
right.
positive, and that's just like that's what it looks like when you run the sample itself.
So I mean, it could be that it's linearized, and so it's all unwinding into this long line, and then you can have like you can have multiple conformations maybe that are more stable than that, but that's just my random guess. So don't quote me on that.
Well, one thing though is that what you're not seeing is like several other bands, right? Like it's presumed that idea is like that is the protein itself, and they get a little bit closer in the on the right hand side. Then they're running gel filtration, I think there, and so then they're getting a size. They'll get an even more precise sizing of these things.
Yeah, I'm not going to pretend to know exactly what's going on here because I mean I noticed that there are 2 bands and there is like a third fainter band like up at the top that's very similar to what you see in the like normal antibody size. So I'm not.
Yeah.
So
I
I'm
mean that is some sort of impurity, I would say, right? But like I guess the thing is it's not that impure, or maybe it's not. Maybe we're wrong, right? Maybe that's
yeah.
not an impurity. It can be explained away in a certain way.
So this is a point where I have to sit back and go, I'm not an expert on this specifically, and I wasn't, and even if like I was working in the same lab as a person, I'd probably be going, I don't understand what that.
Yeah, you can just
Yeah,
ask somebody. Ask an
yeah,
expert. Yeah, we're not. Let's move to the next. But
yeah,
but the takeaway was they tell us that it's a fairly clean antibody that they made.
yeah. Now this looks at the protein stability of the nanobody FC fusions, those bivalent
Yeah.
antibodies they created.
So they really cranked the temperature
Yeah,
to see like at what temperature does it all kind of break apart at. And it's pretty good, right? They can get it fairly high, and you still, yeah, they're stable at a fairly high temperature.
yeah.
The nanobodies and the MABs.
I mean, as long as they're stable way above body temperature, I think that's what the main thing we want to make sure that. And of
Yeah.
course they should be because they're derived from an animal so with a high body temperature. So I guess it,
Yes.
but it's
Yes.
just to make sure.
But you don't know, right? Like
Yeah,
I think it's important you don't know because they put it through that evolution process where like things were just randomly changed, right? You could get that scenario where, oh, it's a good binder, but actually it breaks apart in heat. So like it is important to sort of cover these bases.
yeah,
And then we spoke about this before that because you're going through this system where it's just how well does it bind, you also don't know whether it just binds everything really well. And so here they take lysate from CHO cells and they say that's Chinese hamster ovary cells, right? Sort of a unrelated group of proteins.
Oh,
And does
yeah.
our nanobodies bind this?
I mean, I thought this was human embryonic kidney cells, but
Oh,
uh,
was it? Heck.
heck cells. So, but yes,
I wrote show when I put these together, but might not be.
I mean, I wrote heck 293 in my notes, so I don't know. It could be both, but I
Well, nah,
I
I
don't
don't think it can be both.
I don't yeah I don't know.
This is easy. We can go to the figure. I have
Let's
them open here too.
all right. Let's go to the paper.
Yeah. Let's go to the paper. This is figure 6.
Yep, figure 6B.
Okay. Figure 6 says non-specific bivalent antibodies and nanobodies and antibodies immobilized on magnetic beads were evaluated by incubation biotinylated soluble membrane proteins from CHO cells and detection
Right.
of non-specific binding by flow cytometry.
Right. Okay, that makes sense. Okay. I don't
CHO
know where I got the
cells.
heck. Yeah, CHO cells. All right.
Heck cells are how they make the antibodies, and they
Right.
also did something else with hex cells. I think one of their early binding assays, one of their neutralization assays was. Oh yeah, their pseudovirus neutralization assay was to hex
Yeah.
cells as well.
I must have gotten confused about that, but I mean the important thing is that
Problem.
you've got this mammalian. So again, this is like replicating that immune process where cells are filtered out if they hit the self.
Yep.
Um,
But you
so
know
again,
what?
this
You kind of stumbled onto this thought that like if they used hex cells, then they could say something about human cross-reactivity. But because they use CHO cells, it's like Chinese hamster ovary cross-reactivity.
yeah, I mean, but I guess if they use it in hamster model, it'll be really good. So
Hmm. Hmm.
and they can always do more filtering later to double check. But I think
Absolutely.
that they do like kind of establish that there is low non-specific binding, which is I think a really important thing.
Yes.
Because I mean, in theory, there could be like one unique human protein that's only produced by humans that could theoretically bind to this, but we don't necessarily know, and it could be that protein would be I don't know. No, I have no idea.
I was just thinking,
As-
like, let's say they did find a non-specific binding. It's possible that you could modify your evolution experiment to try to eliminate the non-specific binding, like doing some sort of negative selection onto it.
yeah,
Our bodies do that, right? So, like, there's that possibility as well.
yeah. I
Um,
mean, you could run that fax plot, and one of the axes would be the non-specific binding, and then you'd
mhm.
filter out the things that are very low on that. So you'd have that'd be the kind of I guess in that would be the right-hand lower quadrant they'd be looking at, and then you'd
Yeah.
pick out stuff that.
Exactly,
So yeah,
exactly. So, it's kind of interesting, right, to think that like these aren't even a death knell to like the technology, right? Like if it like had a bad profile in any of these two, like you could be like, okay, not easy, but you could still make these antibodies with a different, slightly different process. But the fact that it worked out so well in this first try, you know, the story that they're trying to tell is it's easy, right?
Mm.
And it helps to show this information.
Yeah, so they do start to do intentional CDR swapping. So they swap CDR regions out between plasmids in order
Yeah,
to.
so they noticed, right, that
Mm,
that it looked like the CDR regions, like some of them were just swapped around, right, when they
mm.
sequenced the original ones. So they're like, yeah, let's just instead of doing the whole directed evolution thing, let's just take that initial library, the one that was made 2018, right, that has all the different diversity in it. Maybe in there we'll find the thing that we want if we just swap the CDR regions. And you know, this is also really similar to the way our bodies generate diversity, right? This is
Yeah,
VDJ recombination.
yeah, yeah.
So once again, just copy nature. They seem to have a lot of things embedded in the systems, right? In terms of how things work.
Yeah. Uh, and that brings us to Figure 7, where they take these antibodies that they created, like just through this swapping, and
Yeah.
they measure up against what the original ones.
Actually, wait, can you go back? I think we should
Yep.
just explain to people that swapping again. So you mentioned it before. You're PCRing those regions, the CDR regions out. So imagine this library. It has all these different combinations of CDR1, R2, and R3, right? If you PCR them all out and you give them little ends, right, that are compatible with the framework, then you can mix it. You can mix them all together and mix and match them, right? You ligate them in a way that you get different combinations of CDR1 and CDR2 and CDR3 together.
Yeah, I guess it almost like, I think the term I've heard used is biobricks. The idea that these like kind of
Biobricks. Yes.
areas that are very similar between these cells are act almost like the cement that it kind of
Yes.
it
Yeah.
very much like they're very similar, so they're complementary, so these will bind. So if you have like these kind of bits of DNA in like a little soup, those complementary ends will start binding, and you won't be able to control how they bind. Those do in random directions, and that will
Hmm.
allow them to swap.
Yep. No, absolutely. That's a really good point. Yeah, biobricks. That term I think comes from IGEM, the
Yeah.
International Genetically Engineered Machines competition. They're really big about trying to like standardize a lot of what we see from engineers trying to understand like how will I engineer this system. And once again, I want to highlight as a biologist and not an engineer that there are lessons in biology that can teach things to engineers in the way things are done, right? In some ways, that's what has happened here, right? Is that they're like, oh, let's try this technique. It seems easier, but that's VDJ recombination. Okay, let's move to the next one.
Yeah, so now they should tell you how they do it, but now this is like the result. So
Yep.
they create new antibodies. I think they're called K719 and K713,
Hmm.
and they
Hmm.
run those up against the things that were created.
Their
And also they
ancestral version and one of the evolved.
yeah, and they all do better than the ancestral versions, like which kind of indicates that the CDR swapping mutagenesis is just a real big value added for creating nanobodies.
Yeah,
So
totally. Which is great. Like so, like it's what an interesting story in the sense of they set out to do directed evolution. We told you a big story about how directed evolution is like you're taking out the body, you get to like look at unbiased way, right? Like a pool of potential candidates that you wouldn't have otherwise seen. It takes all this work. You have to do this iterative steps, right? Pick out the right thing, choose your phenotype, right, really well. But in the case of antibodies, or nanobodies at least, you can also just do this swapping method, which is an evolution step, but you just do it once, and then that generates a good amount of diversity that you can pick out really awesome candidates.
yeah, and I think the interesting thing about this is that while they did use it for SARS-CoV-2, there's nothing stopping them from using it for other things as well. There are
Hmm.
lots of other antibody therapies out there that could potentially be like nanobodies could be a good competitor to them. And
Yeah.
if say someone wants to go back and say create the new I don't know like Herceptin or other like kind of drugs, then they can go back to these
Oh
nanobodies
man, no,
and
the highest selling, one of the highest selling drugs is Humira.
right, yeah,
Is an antibody against an immune target. I don't remember what it is, like IL something.
But yeah,
But yeah, like that's certainly why drug companies are interested in this sort of thing, right? Is that like they make that sort of cell culture, and they probably have troubles with yields and things like that. If they remade that drug in nanobody form, they could get much better binding and maybe higher yields.
yeah, and it could be useful against things that we haven't seen yet, or it could. I mean, this is quite there's it at the moment the limit is like thinking like what do I want this nanobody to bind, and then just running with that because again
But
you can also do
that's also from the therapeutic side. But let's think about like the research side, where it's like I could have an antibody, right, to my favorite target,
yeah.
without having to go through an animal, and I can right, and I can get a better binder, right? I could like at this nanomal. Also remember this. That research you also need you don't need as good nanomolar levels to get
Mm.
to have a good research reagent, right? Like at this level, like yeah, this is great. I could do like a 1,000 dilution, right, with a secondary to amplify that signal, and I could like find yeah, I could get antibodies to these targets. I actually really love that thing, right? This is like a big DIY perspective too. Is that if the technology to make these is easy, right? Like I mean, it's not because this is in a big research lab. Um, Working on making it more accessible is a way to get certain reagents into people's hands who otherwise wouldn't have them, right? Like imagine if you could do, I mean, sounds bizarre, but like what if you did want to do staining for some particular thing in the soil or like in your favorite fermentation, right? Like right now it's like, oh, you got to buy those reagents from some company, but like it'd be cool if you could do it yourself. That's just me. I'm like really
Yeah.
into that thing.
I mean, also like doing things like ELISAs or things like that, where you're trying to do like a biomarker test. That could be quite
Yeah.
useful. I mean,
Rapid
yeah.
prototyping for making different ELISA assays. Very good. Yeah, that's also a very interesting application.
So yeah, I think we this was a really good one. I really enjoyed talking about this one. Uh,
Yeah, no, happy to talk about the paper. I thought it had a lot of. We got to spend more time talking about directed evolution, right? Which, as we mentioned before, is something that we briefly talked about in ACE2 decoys uh, deep dive, and probably will come up again. We also did it with the phage. That was a type of directed evolution,
yeah,
um, and but sort of more like for the whole organism instead of for a specific target. And yeah, um, evolution is a big force, right? That we're sort of controlling both on the ecological scale, but also in the bench.
yeah. I mean, it's
So
going to be an important tool for let evolution do the thinking work for us sometimes.
totally. So yeah, so join us next week for our deep dive week where we will, oh, sorry, for our news week where we'll
Yeah.
survey papers to try to find something that we'll cover in detail the following week.
And we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it is possible we didn't get everything right. So, science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter with the hashtag #MicroTWJC. We both believe that peer review is a process that we can all participate in, and we hope you all have had a good time listening to us ramble on about microbiology today. So if you think you have something to add or you found something unclear, please let us know.
It's been a pleasure chatting with you, Danny.
Same here, Paz.
Tune in next week for more microbiology content. Bye!
Bye.